This is John MacKay, and another session with a podcast, and an interesting one, I think, today, talking about extraction and talking about different organic chemistry and the chemistry of cannabis and the different things that are timely. And so it's called Extracting the Best from Every Day with Dr. John MacKay. And today I'm joined with Dr. Bennett, and from Prescott Logic Technologies. And welcome.
Thank you very much, John. Appreciate it.
We've known each other a long time, but and crossed our paths so many times that we suddenly see ourselves in a hallway sometime and go, oh, you're here. You know, I didn't know you were here and stuff. And it's been good fun. But it gives me a chance to talk with you today and find out a little bit about what you are currently doing and a little bit more about some of the things that are topical. So with that, I throw it back over to you to tell us a little bit about what your company does and a little bit about yourself, I guess.
Absolutely. So Prescott Logic, I started originally in 2014 in Colorado. At the time, it was to make a transdermal patch, which I did pretty quickly and pretty effectively. And truthfully, it was the beginning of a product development path that still is in a lockbox and waiting to be fully developed, but I have some good formulations with that. So, but as fate would have it, I was doing a postdoc for my 7th year and at the University of Colorado Medical School, Anschutz. And I had an opportunity to develop something and, was sort of awakened to how warm companies were to inventors. Obviously, they want to be on the patents, but I could get a lot of material for free, which I didn't know as an academic. Anyway, got the patch developed, and it started giving me some visibility in Colorado. And I very quickly realized that there wasn't necessarily a dearth, but almost an absence of quality medical and scientific thinking in the space. And I saw more of an opportunity than just, launching a patch, I guess. And I became the scientific director of Evo Lab for a couple of years, was there, and then worked for a company called MacKay in Canada, got some experience there, revitalized Prescott in the US and started Prescott Logic also in Canada. Pardon me on the lights. And so my background is I'm an academic scientist, Ph.D. molecular biologist with emphasis in neurodegeneration, opiate receptors, and melanoma, and I'm a bench to clinic researcher. So analytical chemistry to cell culture to translational animal models, usually mice or rats, to preclinical and clinical studies, and actually have a drug patented from my studies with my team, of course, that's made by Sanofi that we repurposed and is in a few different phase 2 clinical trials for Alzheimer's disease. So had a successful, for lack of a better term, scientific career, but was very, very excited to be in Colorado in 2013 at the time that things were starting up.
So yeah, so Colorado to now Chicago?
Grew up in Chicago. Most of the schooling was Florida, then moved to Colorado and been back in Chicago for about 5 years. Started a family.
Yeah, so that's an interesting transition because in 2013, 2014 in Colorado, there was an increase in the interest, and then very few laboratories that were moving towards some of the scientific processes that had to be in place, as well as analytical testing. There were very few of those available even early on. And I think there were probably a handful of companies that were looking at, besides just extraction, for getting products that were in an oil form and then transforming into something else was an early stage. And so you were in that stage early on. So I always thought that an organic chemist is the most amazing chemist on the planet because we had to know everything. But I would say that on your side, you trumped that rather significantly.
I appreciate that. I don't know if it's true. I think I've had to, as a biochemist, go back and relearn a lot of things I regret not paying closer attention to as I had the opportunity in organic and other things. But cannabis has been a great opportunity to be an applied scientist, which really is what excites me more than anything. And I would have never have known that if I was, still in academia.
Yeah, so as you're looking at that process and you see some of the things that interest me and the things that you're doing are taking the compounds and then tracing them through the metabolism of a organism.
Correct. So it's kind of bimodal, and I should add what Prescott does is sell top end purpose-built fractional distillation equipment specifically for the hemp and cannabis space. That's we sort of evolved into that while I was doing product development, started my own company. I realized that people had a lot to learn in the processing world to be able to get to a patch. At the time, a lot just had extracted oil. And so I was reselling equipment, and then my current partner, Pilotist out of Germany, I went to visit them. They came out on day 2 with a bunch of drawings and had my logo and made me OEM. So we're quite proud of our distillation equipment. Just to tie off what Prescott does. Sorry, I'd be remiss not to finish that off.
Yeah, it would be, I guess. So as you're moving through different phases of distillation, which is now you're looking at vapor pressures and you're looking at solubility and separation and seeing that. So as I've always told people, there's a reason why if you go past Richmond, California, or you go past Commerce City and outside of Denver, that they have very large stacks. And being able to do that precisely and accurately is an art. I mean, it's not just putting a couple pieces of glass together and a little bit of cold water in the middle. So there's more to it than that.
Absolutely. And especially for a tricky matrix like cannabis. But I appreciate that our systems are vertical. We get a couple complaints, but every other industry, they are vertical to run the most efficiently and not, pumped up into the still. But I digress. But that being said, distillation is has changed a bit as cannabis evolves, which is fun. It's sort of I like to call it a standardization step, because you're really taking something that is full spectrum, has quality, like that's good oil, and really just whacking out the fraction that you want, ripping away the personality and saying this is the quantity of cannabinoids that you have. But it's less of a craft product, admittedly, but definitely standardized products necessary for gummies and patches and capsules and every other thing that you're making. And furthermore, if you are interested in cannabinoids, plural, beyond THC and CBD, it's the right starting material for preparative chromatography as opposed to crude oil. So there's value to distillation in general processing and also for finished product.
It does bring to mind to me, it was, I'd read a paper, and maybe you've read this too, and I can't remember the actual references. I'll have to find it after the podcast. It was around 1909, the first distillation was described in the scientific journals. Fractional distillation, okay, not distillation, fractional distillation, where being able to separate them, a couple of compounds from each other. And it was, I believe I'm going to go out on a limb, I believe he was from MIT, and he was called the modern father of chemical engineering. And so I found that paper, which, I didn't realize that it was not that old. I mean, it's not that old.
I mean, 1909 is not that long ago at all. I had a grandmother that died great-grandmother died in 1984, who was 107. So born in 1876, she saw everything, the entire Industrial Revolution and everything like that. So no, 1909 is, definitely a couple of generations ago in my family.
Yeah, that's well, will be interesting to see what happens in 2052 when you're still working.
Well,
So
interestingly, I think, it's one of those techniques that won't change a whole lot. A lot of people in the cannabis space think something's new all the time. And as you and I both know, nothing's new in this space at all, except the cannabis itself and our interpretation of it. All of these things have been used in many other industries and stuff like that. So
speaking of that, do you see yourself branching out into other spaces besides just the cannabis market itself?
absolutely. Had a call today with a group that, I've had numerous calls, but a group that I'm going to be starting another company on with psilocybin in particular. A couple of interesting twists to what we're doing, but at the end of the day, I extract, I formulate, so that's no different with psilocybin. I think that there's going to be much more of a push for research, which is something that everyone always talked about in the beginning of cannabis, and no one really did, minus a few groups. I think it was just to get my interest. But I think with psilocybin, I really think that there's a lot that's unknown. I think it's a stronger chemical. I think there's going to be more product liability, stuff like that. But as far as, what I have seen for depression, what I've seen for PTS, and what I've seen for traumatic brain injury for TBI is just kind of mind-blowing. So to, again, rule out something as an effective therapy because it's been used a different way historically, I think that pattern reappears here.
Yeah, I think it does too, and I think it goes back through and goes through the purification process, as well as the typical path for a natural product is to take a natural product, find out what people, the indigenous, population is using it for, and then from there, trying to find out which compounds are actually active, which ones are contributing either by deactivating or activating some process, and then from there isolating those and then deciding which parts are efficacious, and that's the pathway that sort of cannabis sort of leapfrogged over and not allowing the science.
I think there's two. There's sort of the top-down approach where you, like you're describing, where you take something like crude oil and dump it on cells. And then there's the bottom up where you take pure THC and put it on cells, THC and CBD and put it on cells and sort of recreate things, or in mice or in people, in order to get more basic data on mechanism and effectiveness.
So when you're looking at that, and I just bring it up as a topic, and that is as far as the receptor sites and having a lotion on a skin epidermal layer, or having something that goes under your tongue, or having something that's IV injected. As you're looking at those and through your experience as a biochemist, where's that fall into?
Well, very loaded question on your part, but I think that it falls into the not truly proven yet, and not proven with the the means that you and I are used to seeing things proven with. And it doesn't necessarily have to be prohibitively expensive clinical trials, but I just think that we're dealing with a lot of anecdotal anecdotal evidence, and we're dealing with populations that are telling us, hey, I have less pain. So like we're going back through and saying, well, what components could it be? Is it THC? Is it CBD? And it's just kind of a maelstrom, if you will, from a research standpoint. I'm not sure if that answers your question, though.
I think it does. I think, and you provide, in some respects, the company and yourself provide some of the compounds that would be used in that research as well as instrumentation.
So certainly, I mean, instrumentation, experience, often, we're around for more than just an equipment sale. I like to say deconstruction and reconstruction. We're around for breaking it down into purified compounds and then mindfully reassembling those into a delivery mechanism to be utilized in whatever manner we see fit. But going back to your question about maybe a cream and some of these things, I think that, or a sublingual tablet, I think what's really missing for me is the pharmacokinetic data that says, hey, this is our fizzy drink, this is everybody else's fizzy drink, and here's where ours kicks in faster. And even if I saw data like that, I don't know how much I buy it, because we've got pharmacogenomic data that says that you and I could have 200-fold differences in how we interpret drugs of any sort, let alone cannabis. So, I just think it's at the same time, I would, what we understand scientifically and medically, I would never want to naysay someone that said something worked for them. Whether that was real or imagined. If someone says this works, it's a doctor's responsibility to nod their head and say, okay, well, that's great that you have something. It's not, no one really cares in that case with chronic pain or with PTS or some of these things that are relatively untreated. No one really cares about the mechanism. So we can sit here and dissect and whittle away all we want, but people can go buy these things pretty much over the counter and treat themselves.
Yeah, when I'm seeing the same way that I go when I treat myself, when I'm in a drugstore and I look at the 7,000 different choices for toothpaste, and do I want to whiten my teeth, or do I want to super whiten my teeth? Do I want to have fluoride? Do I not want to have fluoride? Do I want to use a left-handed toothbrush, right-handed toothbrush? Am I going to use water pick? Am I going to use floss? What kind of floss am I going to use? And that's just the toothpaste, and that's not even deciding what I'm going to have. So when I'm looking at that, I think that each one of those processes comes in place and speaks to what we do as Americans, certainly, because I'm more used to Americans. I see them almost every day, and so and watch them in the drugstores. But that brings me to even some of the things that people can purchase currently on the market today, that as the market started off with 2013, 2014, you would think that we had just discovered, Delta 9. I do remember in early 2014, the first time I saw Delta 8, and I know exactly where I was. I was actually over in Arvada, methyl. And it was there that I was seeing a hump on the side of a Delta 9 peak. And I'm sitting there going, and I explored it more and then did orthogonal chromatography. There's a shock to you. I did orthogonal chromatography and saw very clearly that it was a separate peak. Now, what was it? And at the time, my concern was that the Delta 9 was being overrated, or over, the potency was too high for what it should have been, and that the Delta 8 was therefore something was coming through the process. And so I was on the other side of saying that you're showing too much Delta 9. You have something else in there. It's called Delta 8. It's not as potent, and so you're mislabeling your Delta 9. And now we've gone 180 the other direction, and people making Delta 8. And I have my opinion on that one, and that is that when Delta 8 comes from the biosynthesized Delta 9, it's in the right enantiomeric form. It comes in as minus trans Delta 8. It doesn't come in as the other 3 possible enantiomers. However, when you do bring it from another product, CBD, for example, and you close the ring, you now have multiple products that can be derived from that very simple reaction.
Mhm.
So people are buying this, and for me, if I do a quick cost analysis just in the top of my brain, it just seems like the COM, the cost of manufacturing, doesn't allow me to get to the product suggested manufacturing pricing. It just seems too low, and too many other compounds in that mixture.
Yeah, I mean, there's no standard of quality. So, we have a solvent-free, very clean method of isomerizing from CBD to Delta 8. No residual solvents, nothing like that. But at the end of the day, we're also left to the testing companies to use HPLC to determine the actual concentrations of these things, which doesn't adequately resolve those two. Obviously, NMR very clearly resolves them, but even GCMS does a pretty good job. Almost an order of magnitude better than HPLC. You'll see a 10% D9 go away and become a fraction of a percent when you go to GC. So there's that to contend with, but the biggest problem is that nobody cares about having a compliant, clean product. Everybody just wants the cheapest stuff. People want it to be clear, even though it oxidizes in short order and turns all sorts of colors. The market demands it, and it's a race to the bottom. And until there's any regulation or quality control, or a gas station that says, I'm afraid of hurting people. I think we have sort of an overlap of, is Delta 8 good or bad? I mean, I think we both argue that most of the production methods for it are bad, especially if not properly remediated. But if we just focus on Delta 8 itself, if we were allowed to, I've had some patients that are highly paranoid from Delta 9 that can tolerate Delta 8. It could absolutely provide another sort of therapeutic dimension. But I think right now it's, how much heptane or hexane is left, and how, it's just, I don't know. It's not a pretty picture, unfortunately.
Yeah, so when you speak about the gas chromatography and being able to have, I'm sure it's capillary, but when you're going back through in that process, are you speaking about FID or mass spectrometry, where you can then go to the NIST database and know exactly what the fragmentation is?
Got to be followed by mass spec.
So for those people listening, when you do gas chromatography and you do the mode of mass spectrometry that's done afterwards, there is an very extensive database that allows you to identify clearly the Delta 8 from the Delta 9 based on the fragmentation program. So think of it like a fingerprint. And when Dr. Bennett speaks of the nuclear magnetic resonance, again, each one of those carbons has a very specific environment that it's in, and therefore it's able to look at its environment. It knows very clearly which one it is. There was actually a good work that was done out of University of Rochester forensics group. It was a rapid communication that showed that every single commercially that they bought a Delta 8 product nowhere near had what was stated in it, and all of them except for one had a significant amount of heavy metals still residually in that mixture. And I think that's what you're speaking to.
Yeah. That's
So,
an example.
as an example. But at the same time, if we go into we'll take a little deep dive into biochemistry and talk about KIs and the fact of how you know that the delta 8 versus delta 9 and how it is received into a receptor and how much is received, and so that I'm assuming you're referring to the fact of how the delta 8 is brought into those receptors and activates them.
Yes, and prevailing understanding now is that effect is a fraction as intense but parallel to delta 9. I'm not sure if that's true, whether that's folklore, whether that's based on the scant amount of scientific knowledge that is out there. I'm not sure, or a combination therein. But yeah, I mean that's.
So when you speak of that, because I read a lot of the brochures and the marketing, and most of them say that there's an enormous amount of work on delta 8, and I've yet to find it in peer-reviewed journals of the enormous amount. There is work that's done on the very specific enantiomer that I spoke of, not of the other possible ones.
Right.
And I think that's the part that has to be known by the public.
Well, and I mean, that's something that people are starting to talk about too, in the analytical world, I should say. But wasn't it you the other day that told me that we recognize 1 when it comes to delta 9, and the plant uses 1 when it comes to delta 9, and that kind of, wasn't that you that said that?
Yes, it probably was. If it wasn't me, it was a very intelligent person with a high.
I love it. I love it. But yeah, so I mean, these issues of enantiomers and diastereomers are going to become more of an issue. As we really start proving what's having what effect.
There was a paper that came out today that I had read, today being June something 16th, and it spoke about the S and R. I think it was S and R of limonene. That would make.
It's really odd that was an analogy I was going to go right to.
You were? Go for it.
Well, no. I mean, it's the exact same type of thing. I mean, we make such a big deal in the terpene world about D-limonene and which exact stereoisomer that we're talking about. They're not the same, and you have your S and R, you have your what are protein designations? They're eluding me. It'll come to me. But your DNL are your proteins. Your sugars have. Anyway, edit this part, please.
We'll do the stop and restart.
Yeah, no, I mean, but yeah, for some reason I can't think at the moment. So
we'll do a quick break. I'll count to 3.
There you go.
When I'm looking at the designation of the enantiomers, there is well-documented research on the terpenes that show whether anti-inflammatory or looking outside of the biofilm of what effect it has between what is called S and R. So that's a rotation of light around a molecule, and they're very distinct. And so you not only have that work, and it shows a very distinct level of which one attacks the biofilm. And so that's important through metabolism. And you also have the work that was done over at the Emerald Conference by, I forget who actually showed that work, and that was as the cannabis plant makes the terpenes, it makes a very specific enantiomer and rotation of light of those terpenes. And so if you just add synthetic terpene, it had better be the right one, because one has absolutely no effect and the other one does have an effect on anti-inflammatory in combination with the cannabinoids or on its own. And so you have a lot of work that's done on that throughout the years. And so sometimes we just get carried away with, oh, well, it's, I've got D-limonene and I'm keeping my terpenes, but they rearrange very quickly.
Oh, I mean, for people that say they want to make stable products like capsules and still have terpenes in them, they're not the same a week from now, a month from now, a year from now. And it's a very dynamic pool of molecules that everybody's trying to pin down profiles of.
So looking at all that, and then looking at some of the different designations, and then in your experience or your crystal ball in front of you, where do you see the marketplace going in the next 3 years? I'll make it just 3 years.
You say the marketplace, are we talking about the marketplace of synthetic cannabinoids?
I did make that a big huge hole. So I will go back through and refocus it. So if I was looking at the marketplace from the biomass to the traditional route of using those natural products without doing further synthetic, I would separate it into that first before I'd head towards the second.
So your next step would be to figure out all these specific enantiomeric ratios of cannabinoids, for example.
From the natural plant.
Right.
Through either gene splicing or some other mode of just natural work.
Yeah, it's interesting, because if you would have asked me this question 5 years ago, I would have thought that there would be a wealth of information by now. We've got a few groups doing sequencing. We probably both know who they are. And it's not as evolved as I know I would hope. I think you and I probably agree on that. There's a lot of science here, both from an analytical perspective as well as an applications perspective, disease states, and I feel like we're kind of floundering. I feel like there's not a lot of, there isn't emphasis put on this by the government, right? It's still illegal federally and all these other challenges and hurdles. But I think that universities would have a field day. I mean, I did high throughput screens to come up with compounds that might have an effect in Alzheimer's disease, right? Thousands and thousands and thousands. So to have the flip side and have a plant that has hundreds of hits and just figuring out where and how those work physiologically is quite exciting. But most people need to make money, and I don't think that's a bad thing. And they just, whether it's the medical or the recreational or both of the local municipality, they need to make money and start covering overhead, and this whole what does it all mean kind of goes by the wayside.
Oh, I like the way you phrased the question. I'm going to have to remember that and not give you credit. So as I look at that, if I was to ask myself that question of 5 years ago, if someone had asked me that question, what would have been my answer? And I can look back and know what my answer was because I wrote so many articles on it. But asking myself that question today, by the pivot of where the industry has gone, was not what I anticipated. I was not anticipating this type of turn in the road. But now that I have this turn in the road, now I had to figure out what to do with this turn in the road. Because before then, if people look up the molecules and they think that it hasn't been synthesized, then they have to look up AM for Alexander Macraeonis. They have to look up, RH for Hoffman. They have to look at HU for Hebrew University and some of the other ones where they were really looking for being able to move the molecules where it was going, what it was doing, and its potency.
Mm-hmm.
And then from there versus the natural product. So in my opinion is the following, and then I'll ask for your response. And so I believe the future would be towards products that are behind the pharmaceutical counter, ones that need a prescription. And then I would see some in front of the counter that would be more natural products and formulations.
Yeah, I see the same, and I think it's really going to be determined by the market. I think with federal legalization, which I do think will happen at one point in time, I think all of these spin-off molecules that we're discussing, Delta 10, Delta 8, I think a lot of the HAC, a lot of these will go by the wayside if people just have regular access to the naturally occurring cannabinoids. So I think that's definitely the direction I see things going.
I have another question.
Yep.
So I will say along the line, as you're doing the distillation and you're seeing that process in place and knowing what other factors are you looking into inline or at line analyses?
No, and I'm going to bookmark that and go back because what you were talking about was what we expect to see in 5 years, and I had a good answer about the markets that drive these things. I think what I see as a product development expert is that a patch is going to be probably one of your least abusable forms. It's not going to smell like cannabis. It's the furthest removed. I think capsules, I mean, things that are medically recognized, metered dose inhalers, those are going to be things that are maybe even injectables that'll be available by prescription. And then I think, like I said before, since all these things are available, I think there'll be another over the counter where you might have a natural health product that has some ibuprofen and some CBD and THC mix. I think that's a direction that we will see things go, where things are standardized and still at GMP, good manufacturing practices for production, but not quite proven like pharmaceuticals. And then I still think we're going to see, flower and concentrates and things that the market likes to see and will continue. So I think that we might sort of be at the apex of product diversity. I think that we'll see some refinement and some product validation. And as you indicated, that would be done historically by the pharmaceutical companies.
And those are not quick.
No,
and they're billions of dollars, and they're 10 years. So in between that lapse of 5 to 10 years of billions of dollars and lots of different phases of that work, we're still going to have the natural components. I also have one more question, then I'll set you free, and that is, we're now looking at a plant that is nowhere near the plant that people speak of that was used for 10,000 years. So when people speak of, well, they've used this plant for 10,000 years, that's why we know it's safe. I'm thinking that isn't the same plant that we have out there today. And so have you looked at any work on some of the indigenous plants or anything that was from before all of this changes? I'll call it from the I'll call it from the 60s or maybe even the 50s, people started to really enhance the plant more and more towards the THC or towards other, I'll call
There's
it.
there's
window.
evidence of like shamans in various places being dug up with high potency cannabis. For me as a biologist, a species is defined by an interbreeding population. And if you put hemp and THC cannabis right next to each other, they breed. So cannabis sativa describes all of the same plant. What we're doing is selectively breeding certain characteristics, ideally. So, you know, I think that it is different in the sense that we've selectively bred, just like dogs are different. But I don't know that necessarily that having a larger percentage of THC is going to be problematic. Maybe because people will just use less of it. So I don't really know that there's a direct correlation. I think that we have a tendency to think of drugs of abuse and to think of this being on that category. I think cannabis is very different. We're not rapidly upregulating more and more and more CB1 and CB2 receptors because its effects are felt through several different receptor networks. So it's not as simplistic as that. So I think that debate could go both ways.
Yeah, so I think so if I look at one, I did have one more question that came from that one, and that is, I like the dog analogy. I love analogies, but when I'm looking at that analogy, do you have a good analogy for how the THC or THCA or CBD or any of the other cannabinoids, for example, and they're not the, call it the endocannabinoids for lack of better nomenclature, seems to work. How I've been battling for a nice analogy that explains the fact that the THC doesn't really have an effect on inflammation. It's what the receptor then goes off and makes.
Well, it seems from my understanding, CBD has more of an effect on inflammation. THC is a agonist. It activates CB1 and CB2 receptors. CBD seems to be a reverse agonist, possibly an antagonist in certain circumstances. So that's, you have this sort of on and off effect. But, we have many receptor networks. I think CBD is a dopamine agonist. I think that both THC and CBD have interplay with the serotonin system. There's just, it's a little more complicated than just this drug, this neurotransmitter system. And I hesitate in that regard because I think we still have a lot to learn. There's no question that dopamine's involved, right? Or people wouldn't enjoy it and have euphoric experiences and that kind of thing. So I just, but I also think, we have in our livers, we have alcohol dehydrogenase, right? And that breaks down alcohol. And, why do we just conveniently have that? Well, I think historically we ate more and more fermented fruit. We didn't have access to the freshest. Maybe if you believe in the hunter-gatherers, men have twice the alcohol dehydrogenase. Perhaps they were out hunting and they got fermented fruit when they got back to the home. But, and then you have a lot of alcohol being bad, right? And so I think there is a case for, hey, now we've got a bunch of THC that exceeds our sort of natural balance. But I also think that if that were the case, this has been going on now illegally for a long time and now legally for a long time. And I feel like they're stretching to find cases to write about. Oh, this kid went to the hospital. It's like, well, his parents gave him their gummies or, they left them out. And they generally put activated charcoal and maybe pump a stomach and watch him for 12 hours. And then they send him home with a couple bags of chips. I'm not trying to be insensitive at all, but no one's died and they've really tried to make these things look terrible. So, it doesn't make me think that the higher potency is really going to be a problem, because higher potency also means higher purity. And to go full circle to what we're talking about, we've got tons of impurities and things that we don't want in these compounds. So I don't view having pure THC as evil, right? I think that the potency purity argument is a subtle but big one.
Yeah, thank you. So if you had one final thought to leave our enthralled audience with, and what you would want to leave as a message today for the people, what would it be? And you can choose the people.
Well, I mean, I want to, I guess, say something a little bit more general than esoteric. But, when it comes to cannabis, when it comes to anything in science, we're figuring it out is perfectly fine. In fact, it gives people like you and me jobs. But at the same time, sorry, these lights keep going off. But at the same time, I really would like to have some more concrete ground to stand on, I think, with some of these things. I mean, does THCV affect appetite? Does CBN make you tired? It's just, I feel like we're in a world of folklore. So, I wrote an article once for Terpenes and Testing about understanding the quality of what you're reading. And I think that's a good take home, is that you and I are both scientists, and you and I can go on with acronyms and big words and things that we have to. But for anybody to understand how valuable something they read, whether it be COVID or cannabis, anything that's in the New York Times that mentions something about a group doing something, they're going to cite the group. Then you can take those names, put them in Google Scholar, pull the article. You don't understand a word of it, that's fine. Look at how many citations it has, Google the impact factor of the journal, and really determine, how popular or how well liked that science is and how relied upon it is, even if you don't understand any of the words. And so I think that while, everyone is, you and I were scientists before it was cool, right? I always say that because, the reality of this is that, it does take a village to get the analytics right in order to do proper clinical trials. And, the fact that we're talking about different molecules or different states of energy of the same molecule is highly relevant. And we can't just be saying we know all this stuff when we don't even know exactly what we have in our hands, even when we think we have pure molecule. And so I just think people should be open-minded to the anecdotal evidence, because that's really something we're relying on heavily. And I think people should be patient with the clinical, and I think people should have more faith in the intermediate science. How primary cells respond, or how mice respond. I mean, we're 65% biosimilar to bananas. So I guarantee giving a bunch of rats a cannabis product, we're gonna learn some things about ourselves. And I think that before we do all these expensive clinical trials, I think that we focus on, smaller step science. And I think that can be an innovative, facet for people doing product development. But it was a little long-winded. You'll probably edit some of that, but that's my closing thought.
No, that was a good closing thought, and I think that's I appreciate your time and all the the time we've had, and all the other outside discussions we've had at different times, walking down a hallway going, oh, I didn't know you were coming. And those are the things that bring us to it doesn't matter what the opinions are. I mean, it really goes back down to facts. It's 2 + 2 = 4, and you're not arguing about 2 + 2 = 5.
No, and I think that you and I have always had a lot of fun having different perspectives on things, and remaining open-minded, because science is, shit, you're right. I didn't think of that. You know, I mean,
That way.
it's not about ego or anything like that. And it's also, you know, it's been nice, John, because as you know, it's for scientists kind of lonely in this space. And I'm surprised to say, because now there's Ph.D. programs for what we're doing, but I don't see a lot of people lining up to be on podcasts, I guess, and talk about their opinions and their experiences.
Well, I appreciate the time. All right, have a great day.
Always
Catch
do the
you at the
same.
next conference.
Absolutely. We'll bump into each other. Thanks, John.
Bye.